Welcome to the deep dive. We're really glad you're joining us today because, well, we've got a fascinating topic lined up for you.
Oh, absolutely.
Yeah.
It's a really good one.
Yeah, so today our sources are drawn from a comprehensive anatomy and physiology text, and our mission basically is to demystify adaptive immunity.
Right, which is it's such a complex topic.
It really is. We want to figure out how your body mounts this like highly targeted immune response and then, actually remembers it for the future.
Because that memory part is crucial. That's the whole key to human survival, really.
Exactly. Okay, let's unpack this with a bit of an analogy.
Yeah.
So, if your innate immune system, your skin, stomach acid, mucus, if that's like a generic castle wall, it works in minutes,
Right,
right?
because blocks things.
Yeah.
Yeah.
But the adaptive immune system, that's more like an elite, custom trained special ops unit.
I love that analogy.
It takes days to mobilize, but once it does, it leaves behind these sleeper agents, these memory cells, for the next time.
And to really understand what life is like for you without that special ops unit, we have to look at cases where it's missing, like David Vetter.
Oh, right. The bubble boy from the 1970s.
Exactly. He was born with severe combined immunodeficiency, or SCID.
And that means he didn't have any functioning B cells or T cells, right?
None at all.
Right.
So without those specific cells, your innate immunity, that generic castle wall, it just cannot handle the everyday microbial world.
So a normal environment is basically lethal.
Yeah, mere survival requires complete, total isolation in a sterile bubble.
Wow. Which really shows you the stakes here. But to understand this elite unit, we kind of have to look at how we even figured out it existed in the first place.
Oh, the history here it's wild. It was a massive 90-year scientific feud.
Yeah, a total rivalry in the 1890s. So on one side, you have Emil von Behring in Berlin.
The humoral camp.
Right, the liquid camp. And he was curing diphtheria, which was just a terrifying disease back then, by transferring liquid blood serum, what we now know as antibodies, from one animal to another.
And he proved that the protection was in the fluid.
Exactly. But then on the other side, in Paris, you have Elie Metchnikoff leading the cellular camp.
And Metchnikoff's experiment is, it's just legendary.
It's so weird. He literally took a transparent starfish larva and pricked it with a rose thorn.
A literal rose thorn from his garden, yeah.
And under the microscope, he watched these mobile cells swarm and just like eat the invader.
Which is incredibly visual. But what's so funny about this, and I get really excited about this part, is how incredibly petty this rivalry was.
Oh, they hated each other.
They absolutely refused to agree. The Nobel Committee actually had to step in and force them to share the 1908 prize.
Like parents forcing kids to share a toy.
Totally. The committee basically ruled that both were right. Immunity has a liquid arm and a cellular arm.
Okay, but here's my question, and this is where I always get stuck. How does the body actually know what shape of antibody to make?
Ah, right. That is the big question.
Because, Linus Pauling had that old theory, right? The instructive theory.
Yeah, Pauling thought the invader itself, physically molded the antibody.
Like pressing wet clay over a key to make a copy. It makes sense intuitively.
It does make intuitive sense, but at a cellular level, proteins don't work like wet clay.
Right.
What's fascinating here is how the paradigm completely shifted in the 1950s. Frank McFarlane Burnett and Niels Journey came up with the clonal selection theory.
And this is huge.
It is. They figured out that the invader doesn't give instructions at all. Your body already contains every possible antibody maker before you ever even meet a threat.
Wait, really? Before you even get infected?
Yes. The antigen, the virus or whatever, just bumps around until it randomly selects the one cell that already has the perfect matching shape, and then it amplifies it.
Okay, but hang on. How is that mathematically possible?
The math problem.
Yeah, because how can the human body pre-pack a specific defense for a disease it has never even seen before? Like, we only have about 20,000 genes in our DNA.
Exactly. We absolutely do not have enough room to dedicate one gene to every single possible pathogen in the universe.
So how do we do it?
Well, it took until 1976 to figure that out. Susumu Tonegawa discovered this process called VDJ recombination.
Okay, break that down for me.
So during development, these specialized enzymes, called RAG1 and RAG2, They just start randomly cutting and splicing different gene segments together.
It's like having a small bucket of Lego bricks,
You
right?
That perfect way to put it, yeah.
You only have a few bricks, but you can build, 10 to the 11th to 10 to the 15th unique structures?
Yeah, 100 billion to a quadrillion unique shapes. It's a number comparable to the stars in our galaxy.
Which is just insane. That means no two people on Earth have the same immune receptor repertoires, not even identical twins.
Nope. Your immune system is entirely unique to you. And this massive diversity is split between two highly specialized units. First, you have the B cells.
The humoral immunity.
Right. And the story of how we found B cells is just hilarious.
Oh, Bruce Glick in 1956, right?
Yes. He was a grad student, and he was working with chickens.
He had surgically removed this obscure little organ called the bursa of Fabricius from these chickens just for some random structural study.
Right. And then a colleague of his happened to need some chickens for an immunology experiment.
So he just borrowed Glick's altered chickens.
He just borrowed them, yeah.
Yeah.
And suddenly they realized these birds were completely incapable of producing antibodies.
Total accident. And that's why they're called B cells for bursa-derived.
Exactly. Even though in humans, they actually mature in our bone marrow.
So, okay, a B cell finds a match, but then it does this thing called affinity maturation.
Yes, and this is
A
wild.
textbook literally calls it a Darwinian tournament happening inside your own body, which I just love. It's inside the lymph nodes, right?
Yes. The B cell starts cloning itself, but it intentionally introduces tiny random mutations to try and make an even better weapon.
It's basically trying to upgrade its own design on the fly.
And only the clones that grip the virus, the absolute tightest, are allowed to survive. The rest are destroyed.
Brutal, but highly effective. But B cells only handle the fluids, right? What about when a virus is already hiding inside your own cells?
That's where the second specialized unit comes in, the T cells, which handle cell-mediated immunity.
Discovered by Jacques Miller in 1961.
Right. And T cells, which get their name from the thymus, are completely different from B cells. They are totally blind to free-floating threats.
Wait, they can't just bump into a virus in the blood.
No, they can't see it at all. The threat actually has to be formally presented to them on a molecular display case.
The MHC, right, the major histocompatibility complex.
Exactly. Every cell basically chops up samples of what's inside it and displays those fragments on its surface.
Like putting things in a shop window.
Exactly. And T cells just patrol around, looking at the windows. There's a real division of labor here, too. You have the helper T cells, the CD4 cells.
They're like the generals, right? Directing the whole response.
Yeah, they secrete chemicals that tell everyone else what to do. And then you have the killer T cells, the CD8 cells.
Hey, executioners.
Precisely. If they see a viral fragment in a window, they latch on and inject enzymes that force the infected cell to just self-destruct.
Okay, but that brings up a really scary thought. If we have billions of these randomly generated killer cells floating around, why don't they accidentally lock onto our own healthy cells and just attack our bodies?
Well, they would if it weren't for an incredibly brutal quality control system called central tolerance.
In the thymus.
Yes. Before any T cell is allowed into your blood, it goes through this rigorous audition. The thymus basically shows all of the body's own normal proteins.
And if it reacts to them?
If it reacts too strongly, or on the flip side, if it fails to see the MHC window at all, is immediately destroyed.
Oh, wow. So most of them die.
The overwhelming majority die in the thymus, yes. Only the absolute safest, most perfect ones get out.
Here's where it gets really interesting, though. Because what happens when we mess with those controls? It sounds like we're playing with fire.
We absolutely are playing with fire. And we learned that the hard way in 2006 during the TGN 1412 clinical trial in London.
Oh, that was a chilling case.
Truly chilling. They gave healthy volunteers a drug that was designed to bypass normal controls and just super stimulate the T cells.
Like turn on the executioners without giving them a specific target.
Exactly. And within hours, all volunteers suffered catastrophic multi-organ failure.
Just hours.
Yeah, it caused a massive cytokine storm. Because all the T cells activated at once, they just release massive amounts of inflammatory signals, basically attacking the body's own infrastructure.
That perfectly illustrates how destructive these cells are when they aren't strictly regulated.
And this raises an important question, right? Because sometimes central tolerance naturally fails without any experimental drugs.
Right, and that leads to autoimmune diseases.
Exactly. And allergies.
Huh.
There's this concept from 1989 called the hygiene hypothesis.
Oh, I've heard of this. It's the idea that our environments are just too clean now.
Right. The theory is that our adaptive immune system evolved to fight off parasites and severe infections constantly.
But now we have hand sanitizer and sterile environments.
So this highly trained special ops unit gets bored, it's understimulated, so it starts attacking harmless things like pollen or even your own tissue, which causes conditions like type 1 diabetes, MS, or rheumatoid arthritis.
Because it just needs something to do. That's fascinating. But, looking at the sources, I want to correct a major textbook misconception here.
Oh, about the difference between innate and adaptive.
Yeah. We're always taught that innate is always generic and nonspecific, and adaptive is always highly specific with memory.
That used to be the absolute dogma in immunology.
But that's not totally true anymore, is it?
No, it's not. Between 2009 and 2011, researchers discovered something called trained immunity.
Right, in the innate cells.
Yes. Innate cells, like natural killer cells, they can actually retain an epigenetic memory after an infection.
Epigenetic meaning like the physical shape of their DNA changes.
Exactly. The DNA unwinds slightly to fight the infection, and it doesn't spool all the way back up tightly. It stays a little open.
So it's primed for next time.
Exactly. Which forced us to completely rewrite the textbooks. The innate system does have a form of memory.
So what does this all mean? Like, why should you, the listener, care about all this intricate cellular biology today?
Well, because these mechanics are saving lives right now. Take the COVID-19 mRNA vaccines, for example.
Right, that's a direct global scale application of this exact science.
Specifically, the clonal selection theory. We are literally handing the body instructions to safely trigger that exact selection and memory process we just talked about.
Without having to actually get sick first.
Exactly. And if we connect this to the bigger picture, you have the 2018 Nobel-winning work on cancer immunotherapy.
Oh, this is incredible stuff. Because cancers are tricky, right? They exploit the system's brakes.
They do. T cells have these physical brakes, like CTLA4, to stop them from attacking healthy tissue. Tumors mutate to essentially pull those brake levers, allowing them to hide in plain sight.
The executioner T cell comes up, ready to kill the tumor, but the tumor just pulls the emergency brake.
Exactly. But modern checkpoint inhibitor drugs, they physically block those brakes from being pulled.
They basically cut the brace lines.
Yes, unleashing the patient's own adaptive special ops team to cure previously fatal cancers, like advanced melanoma.
It's just mind-blowing. So, just to summarize this incredible journey for everyone listening, we've gone from 19th century scientists fighting over starfish larvae and borrowed chickens
to realizing your body randomly splices DNA on a galactic scale.
Yeah, all so you can survive the microscopic world. So now when you hear about a vaccine booster or finding a bone marrow donor match for that MHC display case, you actually understand the mechanics at play.
It really changes how you see your own body.
It does. But I want to leave you with one final provocative thought.
Oh, let's hear it.
So the sources note that measles immunity lasts a lifetime, right? But COVID and flu immunity fades pretty quickly.
Right, because of antigenic drift. Those viruses mutate their shape, so the highly specific memory T cells can't recognize them anymore.
The virus changes its disguise, but wait, since we just learned that innate cells also exhibit that epigenetic memory, that trained immunity, could we use that?
Oh, I see where you're going.
Could the future of medicine involve vaccines that specifically train our generic innate immune system?
Providing broad variant-proof protection.
Exactly. Before the adaptive special ops team even needs to wake up. Just upgrading the baseline fortress.
Upgrading the castle walls instead of retraining the snipers every year. That is a fascinating possibility.
It really is. Something to think about. Thank you all for joining us on this deep dive into adaptive immunity. Stay curious, and we'll see you next time.